Greetings to one and all present here today as participants. We'll start with the session. Uh, so, my name is Artik Singh, coordinator of this session, and on behalf of Scientific Communication, Outreach, and Public Engagement, Parul University, it is my pleasure to welcome you all today's webinar on Introduction to Plasma Sterilization in Healthcare Sector. We are honored to have with us Dr. Tejal Barkhade, Assistant Professor, Department of Life Sciences, Parul Institute of Applied Science and Scope, Parul University. Dr. Barkhade holds a bachelor's and master's degree in microbiology, along with an integrated MPhil PhD in nanosciences from the Central University of Gujarat, Gandhinagar. She also worked as a postdoctoral fellow at the Institute of Plasma Research, IPR, Gandhinagar, where she worked extensively on plasma-based microbial inactivation infection control technologies and plasma nanobiointeractions. Her research expertise includes plasma science and nanobiotechnology in healthcare and environmental applications. She has published multiple research articles in reputed Q1 international journals and is also the co-inventor of a patented plasma sterilization technology, contributing towards innovative healthcare and environmental safety solutions. At Parul University, she is actively contributing towards the development of plasma and nano-based technologies for healthcare, agriculture, microbial decontamination, and sustainable sterilization systems. She also plays a key role in scientific outreach and academic coordination and interdisciplinary research initiative within the university. We are delighted to have her with us today and request you to kindly begin with the webinar.
For me, and I welcome all the participants here, and I am also thankful for the scope for organizing this webinar session today on plasma sterilization. So let's begin with the discussion of plasma sterilization and its application in healthcare sector. So let me share first presentation here. So I think let me know the slides are visible or not.
Yes, ma'am, the slides are visible.
Okay. So let's begin with the today's webinar session on introduction to plasma sterilization in the healthcare sector. It's a four state of matter. And what are the state of matters? Plasma as well. So we always get to know the confusion between the both plasma. So we'll also discuss about the basic concept on the sterilization. For example, what is disinfection, what is cleaning, and what is sterilization. And we'll also discuss about the conventional method or traditional method which we have been using in the healthcare sector for sterilization purpose. And we'll see how it can differ from the plasma sterilization method, how it can be differ from the conventional method which we have been using in healthcare sector or the hospital. And we'll also discuss the mechanism of plasma sterilization, how plasma actually kills or inactivate microorganism and how it helping us for the sterilization and in modern technology. And what are the benefit of plasma sterilizers? And we have already available some plasma sterilizers available in the market. We'll also discuss about that. And plasma application in healthcare sector or biomedical research, and what is the current status of plasma sterilization technology in India or outside the India. So let's begin with the introduction of this particular session. So, in our school time, we have studied about the state of matters. When I was in school, in our school syllabus, we have studied three state of matter. One is solid, then liquid, and third is gas. So solid has a fixed shape and fixed volume. Uh, Particles present in the solid matter are tightly packed in fixed position, and it has a strong force between the particle when we talk about the solid state of matter. When when we talk about the liquid have a definite volume but no fixed shape. Particles are close together but can move in liquid state of matter, and we can see the weaker force between the particle in liquid state of matter. When we talk about the gas have no fixed the shape or the volume. So particles are far apart and moving freely in the gas state of matter. But we have also now plasma as a fourth state of matter, and nowadays in our syllabus, people have added, or the education system has added, the plasma as a fourth state of matter. So today's generation is also studying about the plasma in their school time already. So what is plasma? Let's discuss about the plasma. So plasma is an ionized gas made of charged particles that can conduct electricity. So let's understand how it forms. For example, we have a gas now in environment or in a closed chamber, for example. So this gas is made up of neutral atoms or molecules. Okay. Now these gas molecules, when they get energy, when we supply any energy in form of heat, electricity or electromagnetic energy, they get ionized. Okay. So these gas molecules, when they get energy, they get ionized, and this ionization process uh, convert them into the plasma. So now the gas becomes ionized and forms a plasma. So this is a simple understanding how plasma getting formed in the presence of particular gas. Okay, simple gas, when they hit the energy in terms of heat or electricity, ionization process happens and it turns into the plasma. So plasma is made up of ions and electrons, and plasma has no fixed shape or volume. Okay. So let's understand the what is the history behind the term plasma. So many people know about the plasma in biology. The term plasma in biology. So this word plasma comes from the Greek word which is a plasma, and it has a meaning formed and molded. So plasma means formed or molded. Now in 19th century, says psychologist Jane Evangelista used this term to describe the clear fluid. It's a part of blood remaining after removal of blood cells. Now, in blood, when we remove the blood cells or blood cell from that liquid, now white portion which remains there, it is a plasma. The white color liquid remains in the blood. It is a plasma. Now, this blood plasma contains water, proteins, hormones, nutrients, and salt. So this is what happens in 1922. Physics as well. So this is the second time any physicist or chemist, scientist, well-known scientist, Irving Langmuir has used the same term plasma in physics. When he observed that electrons, ions, and neutron particles inside an ionized gas behaved like a fluid carrying a charged particles. So he has also used the same term for this ionized gas as a plasma. So in 1922, he named the ionized gas as a plasma. And ever since then, plasma scientists often clarifies that they are not studying the blood but ionized gas. So it happens with me also sometimes when I am giving introduction of my plasma research. So mostly people think that I am working in the blood plasma, but it is not always the case. Then I have to clarify them that no, I am working in the plasma physics plasma. So now, during the late 1960s, researchers discovered that plasma had a strong antimicrobial properties. So, in late 1960, one group of researchers has discovered that plasma can be used as a sterilizing agent. Okay, so sterilizing property of plasma were officially then patented in 1968 by Wilson Mehanji. So, this particular researcher, scientist Wilson, has identified that plasma has that property that to kill microbes, and when they patented for the plasma as a sterilizing agent. So we can see here the example of plasma around us. Okay, so for example, the best example of plasma around us, we can see the lightning. So we or everybody must have seen the lightning. So how it get formed, the when strong electric fields in so in our atmosphere there is a gas molecules, air molecules are already present. So when strong electric fields in thunder clouds gives energy to all these molecules which are already present in the atmosphere, knocking the electrons free and creating the plasma. Simple, we have already discussed how plasma getting formed. When any gas molecules gets the any energy or heat, uh, and they ionize and form the plasma. So second example is sun and stars. So how this forms sun and stars? Extreme heat in the core of the sun and stars ionize the gas, which gas? Hydrogen helium, which creates the plasma. And third example is neon signs. So we have also seen this neon sign for the signs for the decoration in any hotel, restaurants, or our home itself. So now these neon signs we can see here on the third picture. They are using neon gas, okay, to form a plasma, and it gives the fluorescent color. So electric current passes through the neon gas in a sealed tube. You can see the tubes in a sealed shape. So gas present in the sealed tube. Now this uh, neon gas which is present in the tube, and electron collide with the neon atoms present in the tubes, ionize them and producing the plasma which emits a bright light. Now we can when the this gas present in the tubes, neon gas present in a tube, it gives the energy and it gives the bright light. Okay, so we have also example. We have seen the plasma TVs. How plasma form in the plasma TVs? The tiny cells in the TV are filled with noble gas. Okay. Now electric charge ionize the gas to plasma, which emits UV light that produces an image in our plasma TV. Then next example is the northern lights. So you must have heard about the northern lights. We can see the northern lights in the Norway country in Europe, which is also called aurora lights. So how it get forms? Now the charged particle from the sun collides with gas atom in Earth's upper atmosphere, ionizing them and creating plasma that produces a beautiful light. You can see the northern lights are very beautiful lights are getting produced, which is also a plasma. Okay. So these are some example. The some example are natural plasma sources, which is already available in nature, and some plasma we can make inside the lab in control environment. So let's discuss about how inside the control environment or inside the lab, we can make plasma in controlled manner. Okay, so these are some types of laboratory plasma we have nowadays with us, with the researchers are doing the research in different kind of plasma devices. For example, vacuum plasma processing chamber. So you can see here the picture of vacuum plasma processing chamber, how it looks. So there is a one vacuum chamber, and we supply the gas inside that vacuum chamber, and it has also the power supply. It has a high voltage. Okay. Now when we inject the gas and we supply the any energy, high voltage power supply, it turns into the plasma. Now here you can varies the types of gas. Okay, you can use oxygen, you can use nitrogen, you can use argon. It depends upon the how how plasma can form the reactive oxygen species or reactive nitrogen species, or it depends upon your application, that what kind of plasma you want, oxygen plasma, nitrogen plasma. So you can vary or change the gas inside this vacuum plasma chamber. Okay, now it has a multiple application of this vacuum plasma chambers. For example, we can use such kind of vacuum chambers for the sterilization. That is our main topic of today's session, the plasma sterilization. So we can use for the sterilization or for the bacterial killing or microorganism killing or surface cleaning or for the seed treatment. Now we can also use such kind of plasma chamber to treat seeds, okay, which can ultimately enhance the seed germination by increasing the water uptake capacity of that seed. Okay, now how plasma getting formed in this chamber? Gas inside the vacuum chamber is energized using electric power and reduced pressure, producing a control plasma. Okay, now other example or other device we can use. Plasma jets. Okay. Now same power, and you can like for biomedical application or wound healing and also the seed treatment. Now the plume coming inside from this plasma jet. Now this plume has a plasma which can be used for the wound healing purpose. We'll also discuss more in next slide how can be plasma jet can be used for the wound healing. Now a high voltage electric field ionized flowing gas, the flowing gas from the tube, the jet, such as helium or argon, producing a visible plasma plume. Okay. The third device we can have radio frequency plasma systems. It has also multiple application in thin film deposition and semiconductor processing. Then we have inductive coupled plasma systems. Then we have a glow discharge plasma system. Then dielectric barrier discharge DBD plasma system. So all these are the device which can be used for different kind of application based upon our experimental need or our application type. Okay, the formation or the source to form a plasma can be changed. Okay, and to form the formation of the plasma, and we can use the different kind of gas to form a different kind of plasma, and the property of plasma can be changed accordingly. Okay. Now let's understand the basic concept or the basic difference between cleaning, disinfection, and sterilization. So I just wanted to have an understanding between all of three before moving to discuss about the plasma sterilization in D. So what is cleaning? So cleaning is nothing. It's just a physical removal of dirt, dust, and organic matter from surfaces or object using soap, detergent, water. Okay. But cleaning doesn't remove any microorganism present on the surface. Okay. While the disinfection, it's a use of in disinfection we use a chemical or physical agent to kill most harmful microorganisms or inanimate objects and surfaces except bacterial spore. Now, here in disinfection, we can reduce the number of the pathogen bacterial cells or microorganism, or but we can't kill the spores. So bacterial spores cannot be removed completely in disinfection, but yes, we can remove or significantly we can reduce the amount of bacterial load present on the surface. While very importantly, sterilization process destroy the complete microorganism or the types of microbial life, including bacteria, viruses, fungi, spores, and then foams present on the any surface. Okay. When I'm talking about the sterilization, it means it's a complete removal of life. Okay. There is a no chance of having single bacterial cell or spore present on the surface, which we are going to treat in sterilization process. Okay, which means complete removal of or complete destroy of all the form of microbial life, including all types of microorganism, whether it is bacteria, virus, fungi, or spores. So we have already discussed what is sterilization. It's a complete removal of the microbial life. Okay. Now, why do we need sterilization process or method in healthcare setting or in our hospital itself? So, very first important problem is we are facing in the healthcare settings is healthcare associated infections. Okay, so how these HAIs means healthcare associated infections are getting formed in the healthcare setting or the hospital. So just I'm giving the example of this scenario. For example, one patient we can you can see here the patient. When this patient goes through or takes any surgical or medical treatment, okay. Now when doctors or healthcare person using those surgical equipments, for example, forceps, scissor, uh, during those surgery, and if those surgical equipments or medical equipments are not properly sterilized before surgical treatment, then the chances of infection are high. Okay, now if those surgical equipments are not properly sterilized, so patient may get infection after the surgery or after the treatment. So there is a very important process before having a or having any medical treatment should be properly sterilized. So again, why do we need sterilization in healthcare setting? First, we have already discussed to prevent hospital acquired infections. Okay, we stop the bacteria spread of bacteria, virus, fungi, and spores, and it also protects the patients from secondary information. Okay, uh, it also ensures safe surgical procedures. Okay, protects healthcare workers, then prevent cross contamination. Okay, cross contamination means to stop microorganism from transferring between patients, instruments, and surfaces. Then essential for medical devices and implants. So sterilization is very important process in hospital and healthcare setting before those instruments or surgical equipments reaches to the patient. Then it also improves the patient safety and recovery, also maintains the hygiene in healthcare environment. Now, there are some common areas or objects which are we should and which must sterilize before using for the any treatment or surgery get happens. So these are the example. For example, surgical instruments, hospital beds and surfaces, endoscope or catheters, gloves, series, etc. All these objects and all the material which are being used in the healthcare setting should be properly sterilized. Okay. So we do have our sterilization methods already in our system, healthcare system. For example, we mostly using moist heat, which is also called autoclaving. For this particular thing, autoclave machines are there. We use autoclave machine for sterilization of the surgical equipments or medical objects. Then we have a dry heat option. We also use dry heat, which uses a hot air at high temperature for sufficient time to kill all microorganism. While in autoclave, we are using steam, uses saturated steam under pressure at high temperature to kill microorganism, including spores. Then third sterilization method is radiations. So we are using ionizing radiation, gamma rays or non-ionizing UV radiation to kill microorganisms by damaging their DNA. Then others are chemicals. In chemicals category, we are using hydrogen peroxide, and other is ethanol and ETHO, uh, ethylene oxide. So these are some example of chemicals we are using for the sterilization method in traditional healthcare system or settings. But there are some drawbacks of this traditional sterilization methods. Now, what are the drawbacks of these sterilization methods? Conventional sterilization methods. For example, in autoclave machine or moist heat treatment for the sterilization, the treatment cycle is long and it is also time consuming. And very important, it is not suitable for heat and moisture sensitive items. For example, I want to sterilize any plastic material, okay, in autoclave or by using the moist heat. I cannot do because the mechanical property of that particular plastic material can be changed. Okay, so this is a very important drawback here. Heat sensitive material we can't sterilize by using the moist heat, and it also can cause corrosion or dulling of sharp instruments. Okay, it requires electricity, water, and regular maintenance, and limited to load capacity, cannot sterilize oil, powder, or sealed items. So these are the drawbacks of using the moist heat for sterilization purpose. Now we have dry heat. It also requires high temperature. Dry heat generally requires very high temperature and long exposure time, high energy consumption, and there is a risk of oxidation and discoloration of metal instruments. There are chances of that, and it is not suitable for plastic, rubber, or heat sensitive material again. So here also we have limitation to sterilize the heat sensitive material because we are using high temperature here. Then poor penetration, not ideal for complex or packed items. Yes, because again we are using high temperature here. Then next is radiation. Now it has also limitation of penetration and for the effective exposure to kill the microorganism, and it is affected by dust, moisture, and shadowing, and high cost of manufacturing. It is also not suitable for all the materials. Some plastic may degrade with the radiation. So this is one very important again drawback of using this as a human being. Okay, this kind of radiations. Then next comes chemicals. So chemicals require long contact time for effectiveness. Residues chemicals. Now these chemicals has this residues which are not good for the environment and for the human being. So these residual chemicals may remain and needs thorough rinsing or cleaning. And it can be toxic, irritating, or corrosive to skin and mucus. Effectiveness depends on the concentration, temperature, and pH. And chemicals not reliable for all microorganisms. Okay, so chemicals cannot be inactivate or completely kill all this form of spore of the bacteria or microorganism. So these all drawbacks or limitation of traditional sterilization methods which we have, so that we need a very innovative approach or innovative option or alternative which can also treat the heat sensitive materials and which has some good benefits for the sterilization processes in healthcare settings or the hospitals, which can be a sustainable also. That's why we have a plasma sterilization method. Now, in plasma sterilization method, we can always sterilize heat sensitive material. Yes, because we are not using high temperature here. It's a non-thermal plasma. We are not using thermal plasma for the sterilization purpose here. That's why we can sterilize heat sensitive material here in plasma sterilization, and it is also toxic chemical free. Again, we are not using any chemicals or any toxic gas to form a plasma. So there is a no question of residues of those chemicals, which can be again harmful after the sterilization method for the environment. And we can give the uniform treatment to the all surface and the objectives, which can be further used for the surgical and medical treatment, and localized treatment using jets. Now, if it is not vacuum chamber plasma treatment, if you want to you want localized treatment to any particular thing or object, then we can use plasma jet. It can be sterilized at the localized place, and also it takes very less time, so time is very less for the sterilization purpose. Maximum we can have a 5 to 10 minutes of sterilization time to for the complete cycle. Then also it we use low temperature and low vacuum processes. So these are the some benefits of this alternative plasma sterilization method we can apply and we can use in our healthcare settings or hospitals. So today we are mainly focusing on plasma sterilization method, but there are a lot of application of this plasma in our modern research times. For example, it can be used for the surface modification and material treatment. Plasma is also being used in biomedical research, for example, in cancer and wound healing. Plasma can be also useful in agriculture research, for example, for the seed germination processes. And plasma is also used in nanotechnology, for example, for the synthesis of nanomaterials or nanoparticles. Okay, but today we are going to stick on the plasma sterilization. That is our today's topic of the discussion, and all other topics can be covered in next session. Uh, so let's discuss about the plasma sterilization. So now you have understood what is plasma. You now you got to know or you understood what is plasma and what is sterilization. So when we talk about the plasma sterilization, which means it is a process that destroy or eliminate all form of microbial life using a plasma. It's a very simple definition. When we combine a plasma and sterilization, so simple, we are using plasma to destroy all form of microbial life. It is a modern alternative to conventional healthcare sterilization methods which we are using, and it is also a safer and sustainable method because we are not using any chemicals. We are not using high temperature here to treat our any objects or surgical equipments in these processes. So it's a completely a safe method or technology, plasma technology, and sustainable method. Now let's understand what happens when this plasma gets treat the any microorganism surface. So, for example, when plasma is formed in vacuum chamber, it treating the surgical objects which we have kept inside the chamber. Now, what happening here? When during the exposure of plasma on those surgical equipments, reactive oxygen species or reactive oxygen nitrogen species are getting formed during the plasma exposure on those surgical equipments. Now, what are the ROS or reactive oxygen species? Those are reactive radicals, reactive oxygen species, UV photons. Okay, these are the radicals. Reactive oxygen species also can be H2O2 species, OH radicals, oxygen superoxide radicals. These are the example of reactive species which are getting formed during the plasma processing when we are treating any surgical equipments through the plasma. Now, these reactive oxygen species gets and binds to the bacterial cell wall. Okay. Now, when these reactive species attacks on the bacterial cell wall, it damages the DNA present inside the bacteria. It membrane also gets damaged through this ROS, and it increases the oxidative stress inside the bacterial environment. So, due to that oxidative stress, these bacteria are getting killed and inactivated. So, all whatever cellular material is present inside the cell envelope comes out, or it can damage. So, protein, lipid structure of all the bacteria are getting damaged due to this reactive oxygen species. This is how this plasma works for the microorganism killing or the bacteria killing for the sterilization purpose. Now, there are some factors which affect the efficiency of plasma sterilization. Okay, so nowadays the research and development is going on, that how we can improve the efficiency of plasma sterilization methods or processing. For example, whenever we are forming the plasma, the gas composition and gas flow rate is very important factor for plasma sterilization method. For example, different gas has a different kind of properties. So if you are using oxygen to form a plasma, it has some other properties. But when you are using nitrogen gas to form a plasma, it has other properties. So we need to figure out which is the best gas composition, what are the best gas composition or gas flow rate can be used for the plasma sterilization method. We need standardization and we need optimization of this gas composition to to form a base plasma for sterilization purpose. Okay. Next comes power and power frequency. Then geometric factors. So, for example, the geometry of your chamber or your plasma sterilization machine is very important. Also, the pressure, then quantity, geometry, composition of medical device also plays very important role. How much load you are keeping inside the vacuum chamber for sterilization purpose, and nature and surface density of the microbes. Okay, so how much number of cells or how much load of microbes is present on the surface of surgical equipments or any surface which you are going to treat using plasma very important. So it is also if density of the microbes are very high, so we need to figure out how much time it will take to sterilize that particular object which we are going to treat. Now, then last comes the packaging. After doing all the processing, all the plasma sterilization method, exposure of plasma, how you are going to pack that particular object which you have sterilized using the plasma. So packaging also very important factor in whole process of plasma sterilization. So these are some images which has been taken from the scanning electron microscope, and which shows that how plasma can be affect the microorganism Staphylococcus aureus bacteria. We have used plasma, DC plasma, for treatment of the Staphylococcus aureus bacteria here. So you can see here the control image which are not been exposed with the plasma, or which are not been treated with the plasma. So you can see here in the control image, the bacterial cell wall is intact and it's not getting affected because we haven't used or exposed plasma here. But after exposing the plasma for the 30 seconds, the difference between the control and the treatment of plasma you can visibly see here. When we increase the time of plasma exposure for 60 seconds, 180 seconds, 300 seconds, means 5 minutes, you can completely see the membrane integrity is damaged due to the plasma exposure. You can see curve shaped morphology has been formed, and broken cellular bodies has been formed by using the plasma exposure or plasma sterilization method. So this is a proof. This proves that plasma is very effective method for the sterilization method. So this is a gram positive bacteria, S. aureus. Now see what happens when plasma getting treatment for the gram negative Salmonella abuni bacteria. So you can see here the control image of S. abuni, which is a gram negative bacteria, which has a rod shaped bacteria. But when plasma exposed to this bacterial cell, the cell expansion or swelling of this bacterial cell can be seen. After 30 60 seconds, then 180 and 300 seconds of plasma exposure. So the expansion of these gram-negative Salmonella abeni bacteria is getting increased. Okay, slowly it's getting increased. The cellular expansion and swelling is happening due to the plasma, and ultimately the cellular components which is present inside the cell, it's which comes out. For example, DNA or any cytoplasmic material is present inside the cell, it comes out. And this is how plasma inactivate or kill the bacteria. So in our lab, we have also performed some experiments on catheters, some surgical equipment. So you can see here. And we have also seen the very effective treatment by plasma, and the bacterial loading of the on the this on any these catheters is getting reduced. So after the plasma treatment, bacterial load within the 10 minutes completely reduced after doing the plasma treatment. So we can say here the plasma treatment for the sterilization of all the medical or surgical equipment can be very effective and can be used for the biomedical. Now, there are some limitations of this plasma sterilization method. What are those limitations? So, as a researcher or as a scientist, we need to have a standardize the protocol for the plasma sterilization. So presently there is a limited standardization per protocols or processes is presently is there, and there is a no universal sterilization protocol present yet. So scientists are doing research in this field, but we don't find or we don't found any didn't find any universal sterilization protocol yet. Still, it is in R&D phase. Still, it is in research and development phase. People are using different kind of gases. People are using different kind of plasma source to form a plasma, and they are doing their R&D in their labs. But we don't have a universal sterilization protocol. We says that yes, in this particular time, the complete area of microorganism is getting killed. So this is one limitation. So this needs good research and continuous research in this field, and also the complex process parameters. So we have discussed the parameters of which can be keep in mind for the plasma sterilization, which we need to standardize again, and it has a high initial equipment cost. Okay, so all kind of plasma, mostly vacuum chamber based plasma systems has high cost of equipment that can be reduced after doing more research into this. So there are some areas where extensive R&D is still needed. Now, understanding of mechanism to kill the microorganism, we need to increase the understanding, or we need more understanding behind the killing of bacteria or the mechanism behind it. So presently we know that from the based upon the literature survey, based upon the experiments, ROS or reactive oxygen species or nitrogen species interaction is responsible for the bacterial damage or DNA damage. But still we need to understand the DNA damage pathways or microbial resistance mechanism through this plasma processes. Then scalability and commercialization. So commercialization is very important. When we have any technology uh, for the healthcare or the hospital sector, commercialization is very important. So this is also very important. Then clinical translation. When we have any technology, it should translate to the society. It should reaches to the society. So we are in phase that we are not giving. We have this plasma technology as for the healthcare sector, but we have not reached upon the stage that we can transfer this technology directly to the hospital sector or healthcare settings, so that they can use this kind of technology for the sterilization uh, processes. Then the effect of plasma parameters. So there is a pressure, power, and gas composition are plays very important role for forming a plasma inside the lab, which needs to explore for microbial inactivation. Then material compatibility studies. So we know already that the heat sensitive material can be used and can be treated with the plasma. So there is also need to study that how many types of material can be treated with this kind of plasma. So in India, there are some major institute which are working towards these plasma technologies. Uh, for example, I can say the Institute of Plasma Research, which comes under the Department of Atomic Energy. This institute is working mainly on the plasma application. For example, cold plasma processing, plasma medicine, then of course Baba Atomic Research Center, then some IITs in India are actively working in this plasma research. So you can see here some example of plasma system which we it's present. So for example, you can see here this is a plasma jet system. So this plasma jet can be used to treat the diabetic foot ulcer or the skin wounds. Okay, the plasma coming out from this plasma jet can be directly treat the wound on skin. Here also you can see the plasma jet. It's directly useful for the wound healing. And there are also different kind of plasma system that has been can be researched and can be studied deeply. So these are the some machines or instruments which are available in the market. For example, you can see here the mobile plasma sterilizer. So mobile is the one thing which is there with us all the time. So it has multiple numbers of microorganism present on the mobile screen or mobile surface. So you can directly put your mobile into this small sterilization cabinet. You can see here, and you can sterilize your mobile. And then, for example, the disinfecting or sterilization machine in hospital or environment. So this kind of instruments or machine we have already, but not in the India. These all are machines are available outside the India. And for example, this steroid machine, plasma sterilization machine, which is also there in the market. But the main problem is they are using H2O2 gas, not good directly. The residue of this H2O2 gas are harmful again. So we need more research how we can make sustainable plasma for sterilization methods. So at Parul University, we are also now focusing on the application of non-thermal plasma for healthcare, for the agriculture, and other applications. So you can see here in the picture, we have a two plasma system with us at the Parul University. One is DC vacuum plasma system, other is plasma jet. And these both plasma system are placed at the Lakshya 2047 2047 new building. So if you have any doubt or any uh, collaboration kind of thought in your mind, you can surely reach out to me to the lab, and we can discuss further. Yeah, thank you. Yeah, over to you, Atik. If you have any question in your mind or any doubt or any query, or you want to add something, so you can please ask any question, or you can add something as well if you have anything in your mind. So we have 13 participants here, I can see.
Thank you for your enlightening session, ma'am. We really got to know a lot about plasma sterilization and its uses. Uh, I would request the participants to raise hand and they can ask their questions from their end.
It's not necessary that it should be a question. You can add some points into the in my talk, or you want to add something, or you want to just comment, that is also fine. So I can see the 13 participant here. So those who wanted to join for this particular session, and unfortunately those who are not joined due to other reason, they can see this recorded session at the YouTube. It will be available on YouTube Scope YouTube channel. Yes, I can see the hand raised by the Mukul, Dr. Mukul.
So good afternoon, ma'am. Nice.
Good afternoon. Good afternoon.
Yes, it's a deep about the plasma research. I want to know, is there it is possible that we can able to sterilize water as well as the glass bottles inside our university premises? We can develop certain set of thing inside that using the plasma in a small compact system.
Yeah, if you can see the same slide, can you see this?
Yes, yes,
Yeah, so here you can see the plasma jet.
yes.
So this plasma jet can be useful for the water treatment, and we call it plasma activated water. So we can, of course, treat water by using these kind of plasma jet machines, and then the plasma activated water can be useful for the many application. For as a disinfectant, we can use that plasma activated water, and in agriculture also we can use that plasma activated water for hydroponics or for the seed germination processes. And also you have asked about the glass surface treatment. We can surely use this DC vacuum plasma system, which again you can see here in the picture. We can use this machine to treat glass surfaces as well.
And it is cost friendly because that is most important, because nowadays we are using UV.
Yes.
So it is cost
Uh,
friendly because high voltage and high electricity is required.
yes, you are right. We need high voltage for this particular system. It is not that much it's not, we can't call it cost friendly, but if we'll do more and more R&D into this, then I think the demand will be also can also be decreased. But because we are not going into deep for this plasma research in India, yeah, there are many other institute like IIT, IPR are working, and they are trying to develop cost friendly machines or instruments there. So hopefully we hope that in future we'll get cost friendly plasma system with us.
And the last question is the type of gases that we should use in the plasma system.
Yes.
You have given us the list of gases that, and
Yeah,
we
yeah.
have to standardize the things. Uh,
Yes.
this
Yes.
can also be decided that which gas is cheaper, so that the cost friendly equipment can be made for the sterilization purpose.
Right, right. As I said, gas composition and gas flow rate is very important aspect for the plasma treatment or the sterilization. So I think oxygen and nitrogen and argon gas are kind of gas which we get very easily in our lab. So still we have also demanded for the university for all these three gases. And surely we are going to start experiments by using all these three gas and composition of all these gases for plasma sterilization processes.
And the last thing, that compared to UV radiation, that it won't able to kill the spores, plasma can able to kill the spores. So plasma is more, we can say it's a future for sterilization.
Yes, yes, because mainly many people are doing research on bacterial species.
Yes.
We the on the viruses and other microorganism types, the studies are needed. So research, there are a lot of R&D is needed to see how this plasma can kill all microbial types or microorganism array, the complete microorganism array. We need to study that. So that is also one other aspect that research is needed.
Thank you, Tejal ma'am, for this wonderful session.
Yeah, thank you, thank you. Any other question or any comment?
Thank you, Dr. Mukul sir. I hope Tejal ma'am cleared your doubt. And now the panel is open if anyone has any more doubts.
So this recorded session will be posted on the Scope YouTube page, right, Artik?
Yes, ma'am. The recorded session of this complete webinar will be available on our Scopes YouTube channel, and everyone who were unable to join us today, they can access those and gain the information that you have shared today with us.
Yeah. Yeah.
So if
So
there is no questions, then I would thank you, ma'am, and we will end this
thank
webinar.
you. Yeah. Yeah. Thank you for having me here for the this today's session, and I enjoyed giving this webinar. Yeah. Once again, thank you, Artik and Scope team for organizing this wonderful session on plasma sterilization.
Thank you so much, ma'am. It was a pleasure to host you, and it was an honor to be learning and gaining information from you. Thank you, ma'am.
Thank you.